So this class is going to be a 8-week course, and it is going to go every Tuesday. Now, you can do the work whenever you want. All the lectures are recorded. If you want to find a recorded lecture, just go to the ODIN channel, and they should have a list of the previous lectures for the course. Just click on those and watch the dates you missed. So this class, it should keep you busy the whole time, and it's going to be a much more difficult class than 101 for those who took 101. So be ready to ask a lot of questions, and don't be afraid to ask questions when stuff goes wrong. Uh, you know, reach out and try to document things as much as possible. So when you run a gel electrophoresis, take a picture of your results. When you are growing up cultures, take a picture of the cultures. When you are doing transformations and putting things on plates, take pictures of it. This helps you keep track of what's going on, and also it helps us help you troubleshoot what's going on, so that we can help you, you know, get the most out of your experiments and get them correct. So what we're going to do in this class is we are going to learn how to edit DNA, and we are going to learn how to purify plasmid DNA. We're going to learn how to send that DNA off for sequencing, and we are going to learn how to design our own DNA from scratch. So if you have any projects in mind of organisms you want to engineer using certain genes, this class is the class for you. You're in luck. Um, so you should have all the supplies in your kits. If you haven't received your kit or you just ordered recently, don't worry, it's on the way. It'll be there. You'll have plenty of time in this class to complete the experiments. Um, so just don't worry too much about that. Try to follow along and understand what's going on in this lecture. The first week, I don't think there's any hands-on experimentation. That should start in the second week. The first week, we're just going to try to learn what's going on so we know what's going on when we start to actually work with this stuff. All right? If you haven't already, download Snapgene Viewer. You don't need to pay money for Snapgene. Just download Snapgene Viewer. What this will allow you to do is it allows you to see DNA. Um, it allows you to, look at plasmids and other things in an easy human readable format. And it annotates it for you so that you can really understand what's going on in DNA. Right? Download that. There's a link in the Google Classroom, or if you don't have access to the Google Classroom, just search the internet for Snapgene. Right? So we're going to get started, and there is really no text for this class because all the stuff we're doing is hands-on. So if you have questions or things like that, what you're probably going to do a lot of is read Wikipedia or other articles on the internet to understand what things are going on, but there really is no text for this stuff, just because nobody's written one for anything like this. Gel electrophoresis box. Jin says the kit didn't come with a gel electrophoresis box. So depending on which version of the kit you ordered, what you should have received is a Tupperware container, and all you need is a Tupperware container and tin foil to run gel electrophoresis. That's it. So Gel electrophoresis isn't hard. We'll talk about it. We'll go over it, I think maybe in the second or third week of the class. So really all you need to run gel electrophoresis is a couple 9-volt batteries and a Tupperware container. It's really that easy. Yeah, we'll go over it later in the class. If you want to look ahead and see what's going on, that should be okay. Any other questions before we get started? Anything else anybody wants to know about? All right. All right. So today we're going to talk about plasmids. Now, when most people think of DNA, they think of genomic DNA. And what genomic DNA is that inside, say we got uh, uh say we have a bacterial cell with this little flagella. Inside that bacterial cell, there's some genomic DNA, and it's really big, right? You're talking millions of base pairs. That's what most people think when they think of DNA. So there are other kinds of DNA. One of those kinds of DNA is called plasmid DNA. So plasmid DNA is circular DNA. Usually plasmid DNA is between approximately 2,000 base pairs and something like 50,000 base pairs. If you see the representation KBP, that just means kilo base pairs or thousands of base pairs. So 2 KBP is the same thing as 2,000 base pairs. So these pieces of DNA are relatively small when you think about it, right? If genomic DNA is millions of base pairs, these ones are only thousands of base pairs. It's pretty small. But the reason they're small is because generally we use them to put new DNA inside cells. So in this class, we're going to be working primarily with bacteria, but generally when you do genetic engineering, you use bacteria a lot to make the plasmid and grow up the plasmid, but you can use that plasmid then in any type of cell you want, basically. So if you want to engineer mammalian cells, if you want to engineer plant cells, if you want to engineer reptiles or whatever, you can use plasmid DNA. Now, plasmid DNA originally comes from bacteria. So bacteria naturally have plasmids inside them that they use to store information and pass them on from different generations. The thing is, generally these plasmids don't necessarily stay with the bacteria, right? So if you're a bacteria and you're just swimming around and you got this plasmid inside you, well, when you replicate and you start to become two bacteria, you might not replicate this plasmid because there's no reason to. It costs energy, it costs resource. Stream cut out. All right. Sorry if the stream cut out. I don't know why that cut out. Anyway. So, generally, what antibiotics are for is they are for killing any sort of organism, right? They're antibiotic, which just means against life, right? Not antibacterial, which is different, or antifungal, So there are antibiotics, things like G418 or pearomycin, which can kill human or mammalian cells. G418 works against almost any type of cells or organisms. Also, things like hygromycin, which will kill plant cells, right? And the general antibiotics that we use with bacteria, such as ampicillin, kanamycin, streptomycin, and a lot of others. It just really depends on what you're trying to use. So almost every plasmid you see is going to have some sort of antibacterial selection and an antibiotic selection. Now, bacteria are great at replicating plasmids. This is because bacteria grow really fast, right? So if you grow up some bacteria, E. coli, they have a doubling time of around 20 minutes to an hour. So every 20 minutes to an hour, they're going to double, and the plasmids are going to double. And there's not usually only one plasmid in cell. Sometimes the cells have lots of copies of the plasmid. So you can imagine if you grow up, say, 50 ml of bacteria, and each bacteria has a ton of different plasmids for it, you're going to be able to purify a significant amount of plasmid. And that's what we're going to learn, one of the things we're going to learn to do in this class. How to use bacteria to replicate plasmids so then we can use them for downstream applications. Because basically, that's the goal, is to get these plasmids replicating so that we can use them in some way, right? You can engineer bacteria if you want, or any other organism. Now, plasmids are DNA, and so they're double-stranded, right? 2 strands of DNA. And DNA is charged. So DNA has a phosphate backbone, which has a charge. So when you're trying to put it in the bacteria, it can be quite difficult. You can imagine that the membrane of the bacteria has kind of evolved to prevent things from getting in, including DNA, and especially larger things. So that's why we tend to keep plasmids around 2,000 to 50,000. The average plasmid size is probably about 5,000 base pairs. But it really depends what you're putting them in. Plasmids that go in human cells or other mammals or plants are usually a little bigger, and they might be like 8 to 10,000 base pairs. But once you start getting above 10,000, 15, 20,000, the plasmids become really much more difficult to get into the bacteria to replicate and make. So instead, options are to make multiple plasmids or to try and insert the things in the plasmids in the genome. But plasmids themselves, you generally don't want larger than, like I said, like 15,000 base pairs or 20,000 base pairs. It just becomes hard to replicate and use. Okay? So, are there any questions that you have about plasmids so far? SnapGene is free, Vincent. SnapGene is free. Get the viewer, the SnapGene viewer. Don't get the SnapGene program. Get the SnapGene viewer. It's free. It should be free to look at DNA and do all sorts of cool, interesting stuff with it. Now, so the first experiment you're going to do in the class is grow up bacteria, purify plasmid DNA. So you're going to grow up bacteria in a media that contains a specific antibiotic to select for a specific plasmid, and then purify that plasmid from the bacteria. That should be next week. All right. But right now, what we're going to do is we're going to do a little snap gene tutorial and start to talk about plasmids. I think I got way too many plasmids open here. All right. So this is SnapGene. So if you can and you want to follow along, open up SnapGene on your computer. Just click on your start icon, type in SnapGene, bring it up, and you'll get a screen. And it'll ask you a bunch of different things, and choose that you want to try to import DNA from AdGene plasmids. So try to import some DNA. Now, if you go to the Google Classroom, there should also be some plasmid sequences there that you can download, and you can look at those. But here, you can use AdGene just to add the plasmids that you want in. So get that going, pop that up. Now, AdGene is just a repository of plasmids and plasmid information. So like, usually they only send plasmids to nonprofits, but you can get plasmids to them for $60. What I recommend is if you wanted to get a plasmid or something from AdGene, find a local biohacker space and convince them to sign up for AdGene and have them order plasmids for you. That's usually a pretty good method to go about. AdGene usually isn't too strict on who they send DNA to. AdGene is a great resource with a ton of plasmids that they are available generally for non-commercial use, quote unquote. I mean, yeah, take that as you will, you know? Nobody's going to break down your door and tell you that you can't use it, and nobody's going to sequence the DNA you're using if you plan on doing commercial stuff. It's just some stupid bullshit. Anyways, if you don't get stuff from AdGene, who has pre-made plasmids, there's a couple other places that you can get pre-made plasmids. If you go to atum.bio, what they have is they have some plasmids, some standard plasmids that you can use. They have a bunch of different colored plasmids and a bunch of different fluorescent plasmids. So if you want to get fluorescent proteins for bacteria or yeast or even human cells, you can find them at atom.bio. They're a lot more expensive, usually in the hundreds of dollars, but you can get them. But besides that, basically you're building your plasmids from scratch. So one of the plasmids we're going to work with in this class is P-Dusk. So what you can do is select AdGene and open P-Dusk from AdGene, import it in, and let it pop up. There we go, P-Dusk. They call this picture a Plasmid Map. Basically, this is a high-level view showing all the different things in a plasmid. It's a map of the plasmid. Now, all these different little letters and numbers are what they call restriction sites. Now, restriction sites are where restriction enzymes bind and cut, also known as restriction endonucleases. Let's go over this really quick and talk about restriction endonucleases. Because it's something that you should know about and we're going to use in this class. All right. So generally, you have your plasmid, right? You got your double-stranded circular piece of DNA. And what you want to do is you want to put a gene in this plasmid so that this gene is expressed and made into a protein or whatever you want inside the cell. The question is, how do you get your gene inside the plasmid? Now, the main way that scientists do that is that they cut the plasmid using restriction enzymes, also known as restriction endonucleases. So, restriction enzymes, what they do is they recognize a base pair sequence. They're little proteins, they're enzymes that recognize a certain base pair sequence and cut at that specific sequence. So what scientists have done is they have meticulously composed plasmids that contain these sequences in them so that you can use them to cut, right? So for instance, we could check here, let's just go on SnapGene. So we go to the sequence. So for instance, the ECO R1, ECO R1, we call it ECO R1. What it does is it cuts at GAATTC. Now, a lot of restriction enzymes cut at palindromic sequences, which are the same forward and backward, right? So, ECO R1 is GAATTC. So this on DNA is the 5' to the 3'. Now, on the bottom strand, the sequence is GAATTC. And this is the 5' side, and this is the 3' side. Right? So you get it? So it's palindromic. It's the same on the top strand and the bottom strand, going forward and reverse. Now, as you saw in the picture, restriction enzymes cut generally with what's called an overhang, a lot of the ones we use. So what they do is it'll cut right here and over here like that. So what it does is it removes all this stuff from the DNA sequence. So you have this overhang here. So other DNA can come in and bind here so that you can reform the plasmid. Now they call that ligation. When you bring the plasmid back together and remake the circle, you're ligating things into the plasmid. So if we had a plasmid like this, and let's say we put in 2 ECO R1 sites and cut here and removed all this, then what we could do is we could take a piece of DNA that matched up and ligate it into the plasmid with whatever we wanted in here. How do you do that? You can do that either by getting the DNA synthesized or using a technique called PCR to amplify regions of DNA. Here's the thing. Because these sequences are identical, what would happen if we had 2 ECO R1 sites on both sides is that our new piece of DNA it could go in either direction. So if this is the 5' end and this is the 3' end, it can either go 5', 3', or it can go 3', 5'. So we don't want to use two of the same restriction enzyme sites for each side, because a piece of DNA that also has matching the same site on each side, it means that our piece of DNA we're trying to put in with EcoR1 sites on each side can match in either direction. And DNA is usually directional, right? So what we want to do generally is we want to use two different restriction sites. So say EcoR1 and Hindi3. We use Hindi3 instead on one side, and now if this one is Hindi3 also, the DNA can only go in one direction, right? Because the EcoR1 site does not match the Hindi3 site, and the Hindi3 site does not match the EcoR1 site. But the Hindi3 site will match the Hindi3 site, and the EcoR1 site will match the EcoR1 site. So we will get directional DNA insertion, right? It's just like they're different puzzle pieces. And these puzzle pieces have certain shapes. So we need to choose the correct shape that fits together. All right, does that make sense? Any questions about that? So the cut strand can rejoin, but the thing is that it won't be attached, right? So DNA can bind to each other, right? So if we have like, let's see, where's SnapG? So if we go to this EcoR1 site, if we have two sequences of DNA that are complementary to each other, complementary means that they are opposites of each other, like in this sequence, GAATTC on the top and CTTAAG on the bottom, they will bind to each other, but they won't stay bound. So even if you heat them up slightly, it will cause the DNA to dissociate, right? Because they're only being held together by really weak bonds called hydrogen bonds. Now what we want them to do is we want to hold them together with covalent bonds, right? Which is normally in DNA. The DNA backbone is all stuck together with covalent bonds. So what we do is we use something called DNA ligase. And DNA ligase will stitch the new piece into the backbone and connect it so it's like it was part of the original plasmid, right? So it's like, let's say we have a piece of DNA and we have GAATTC. Now these are all connected by covalent bonds to each other, right? With the phosphate backbone of the DNA. Now what happens is if we use a restriction enzyme and cut out this, right? And then we put in a new piece of DNA that is AATTC. Now these are all connected to each other, but this one is not connected to this one, right? So what can happen, even if we have, when it's bound to the complementary DNA, this can just float away, right? Because it's not actually attached. While this is attached to the rest of the plasmid, and this is attached to the rest of the plasmid, this isn't attached to the rest of the plasmid. So DNA ligase, what it does is it uses ATP to connect this, boom, boom, so then it becomes part of the plasmid DNA. So back to the plasmid map. So you can look at this plasmid map and you can say, wow, holy shit, there's a lot of restriction enzymes, right? And they give you a lot of choices. Yes, they give you a ton of choices. This is because sometimes restriction enzyme sequences naturally occur. And they naturally occur inside the gene you want to put in. So if, say, you have a BGL2 site in your gene, well, you could just use XBA1 instead. Or if you have XBA1, you could use NDE1, and so on and so forth, right? Now, when you have all these different restriction enzymes, usually they put them all in a very small area. They call this the multiple cloning site. Now, cloning in molecular biology is different than when we think of cloning of animals and organisms. Cloning in molecular biology is putting genes in a plasmid and replicating it. So when I refer to cloning, or when you read cloning in relation to molecular biology, it's all about putting genes in a plasmid and replicating it. I know it's weird usage of the term, but that's what it means. So now let's look at the different parts of the sequence of this plasmid. There's a couple main parts that we'll focus on. One, the antibiotic resistance gene. Remember when we talked about antibiotic resistance and how all plasmids have some form of antibiotic resistance, so that we can keep the plasmid inside the cells and select for it? Well, this one uses kanamycin, CAN-R. CAN-R just means kanamycin resistant. So each plasmid, almost every plasmid in existence, will have an antibiotic resistance gene. The next thing almost every plasmid will have is an origin of replication. Now, an origin of replication is what tells the E. coli or organism that it needs to replicate this plasmid just like it replicates any other genomic DNA, any other DNA that's in the cell. So basically what we're doing is we're tricking the cell into thinking that this plasmid is part of its own DNA so that it will make more of it, right? So it's going to hopefully make a bunch of our plasmid because we have these origin of replication. There are other genes that are involved in the origin replication here and a lot of other stuff. So let's look at the sequence to get a close-up of what's actually going on. So here is the around our, where's our multiple cloning site? You can tell a multiple cloning site is because there'll just be a lot of restriction enzymes. Where is it? Right here. We got a lot of restriction enzymes right in here. So now what you're going to have in your plasmid is you're going to have all different things. The good thing about SnapGene is if you don't know what something is, you can just put your mouse over it and it'll give you a description and it'll kind of tell you what's going on here. So you can't just put a gene into plasmids, right? If you remember from 101 or some stuff that you learned before, you need certain things in order to be able to express proteins and genes. These are things like a ribosome binding site to be able to make the protein. You need promoters, you need terminators, you need different elements to be able to make genes. Now, generally, when you're using a plasmid that somebody else designed, you don't have to worry about that. If you insert something into this multiple cloning site, they already have the promoters and terminators and everything like that already there. So all you have to do is put your gene of interest in, and it'll work in bacteria. It'll be made in bacteria. But these different promoters and terminators and things like that are different between every organism. So let's say if we were looking at a mammalian plasmid, now instead of the promoters that are used in bacteria, it uses different promoters like the CMV promoter and CMV enhancer. It also uses different terminators. It uses a BGH polyase signal and things like that, right? So you can't take a plasmid that's meant for bacteria and use it in a human cell and it'll make the same gene. Or plants or fungi or all the different things. Generally, you're going to need to find a plasmid that has the elements that you're trying to use for the organism you're trying to use. Now it shouldn't be that difficult to find a standard empty plasmid. So empty just means it doesn't have any genes in it for every organism, right? They're pretty standard and they're pretty inexpensive. You could find them a lot of places online. And then you can insert your gene of interest into any of these plasmids. So if you want to find a mammalian plasmid, you can find it. And it also has similar multiple cloning sites, right? So it'll also have all the different restriction enzyme sites where you can insert things into. Now generally, if you can afford it, it's usually not that much more expensive to order the DNA than to order the DNA and have them put the genes in a plasmid for you. Now, for instance, no, GMO Rollicus, you can't really bind ends of DNA together that's a gene because there's something called persistence length. A plasmid needs to be a certain size in order for the two DNA to bend and make it come together, right? You can think about it like you can think about it like when you're trying to tie a knot and you don't have enough string, you're trying to tie a knot around something, you don't have enough string, and you just can't get it because you can't get the ends to go through the knot. Well, that's basically what persistence length is like. It's the plasmid DNA only bends so far. Like the DNA only bends so far. As you get longer, it can bend further and further because it becomes less and less, it becomes more and more floppy. But you need a certain minimum length. I don't remember exactly what that is, but and if you've been following along, you're going to need things like origin of replication. You're going to need things like antibiotic resistance, so it doesn't really matter. You wouldn't want to do that. So if you look at website of places like GenScript, so GenScript, what they do is they will do custom gene synthesis, right? So they will synthesize a gene for you, and they will put it in a plasmid of your choice, right? And they'll work with almost anybody. And depending on how long you want it to take, it'll probably take up to 2-3 weeks, 4 weeks to get this done, but you can get it done. You should check it out. They allow custom gene synthesis, and they allow you to select any plasmid. So if we click on it, we can go check it out. So right here, for instance, you can choose all the different plasmids that they allow. So pcDNA 3.1 is a mammalian plasmid, so is PCMV. PET, PET, P blue script, these are bacterial plasmids. They have yeast plasmids, P pic. They have a lot of different plasmids, right? So you can basically have your gene put in any one of these plasmids that you want. They have you define the restriction enzymes if you want to choose the restriction enzymes that we just went over for the 5 and the 3' end. If you don't, they'll just choose it for you, but it's pretty simple. Basically, all you do is you copy and paste your DNA in here, and they will make the gene for you. Put it in a plasmid. That's pretty cool, huh? All right. So hopefully you have a basic understanding of what a plasmid is. Now, next week, what we're going to do is we're going to talk about plasmid purification. So, and you're going to do plasmid purification. So you're going to try to grow up some plasmid in bacteria and purify it using what they call a mini prep or a mini prep kit. Before then, read up on plasmids, read up the mini prep protocol so you're ready and you have some questions. And if you want to know anything between now and then, just let us know. Post in the Google Classroom. Kent, no, there's no organisms for which is preferable to order a plasmid with the gene added. I mean, all of them are more preferable. Genetic engineering, putting DNA together is something that can be annoying at times. You can imagine moving around these tiny amounts of liquid and you have no idea what's going on until the end. So I would suggest, like I said, if you have the money, what you should do is you should order the gene in the plasmid DNA. Usually it's not that much more expensive, So if you look at it, what you're paying is about like 20 to 30 cents a base pair. And they'll put the gene in a plasmid, right? So if you have 1,000 base pair gene, it's $200. Usually genes are around 1,000, right? So there's 3 DNA bases per amino acid, and usually you're about 300 amino acids, So you're talking $200 to $300 to have them make it for you. That's it. Yeah, so it's not too bad at all. Generally, doing it by yourself, you have to purchase all the restriction enzymes, right? The DNA ligase, you have to do all this stuff. So you're probably not going to save that much money anymore. But it's good to have an understanding of the process and how all these things work in case you did want it to do this way. Or if you were trying to clone multiple genes at the same time, and so it really did save you a bunch of money. But as you'll probably find out in this class and see, these things can be quite annoying. No, GMO Rollicus. Plasmids without ORI and antibacterial resistance aren't mini circles. Mini circles are a specific patent protected piece of DNA that is made using a specific method. Generally, mini circles don't have origins or antibacterial resistance genes, but plasmids without origins or antibacterial resistance genes are not mini circles. All right. Any more questions before we finish up? All right. So there are no more questions. Like I said, if you think of anything, post in the Google Classroom. You can find me on Facebook. You can find me anywhere, and I'll try to do my best to answer your questions. Otherwise, we're going to keep doing this on Tuesdays at 4 p.m., between 4 and 5 p.m. So I'll see you again next week. And so, Tom, when you amplify in PCR, um, you're generally no GMO. David Ishii did not reverse engineer one by taking away the origin. Tom, PCR and replicating plasmids are two different things. You can't really use PCR to replicate plasmids efficiently. Um, PCR can replicate genes well and things like that, but generally not plasmids because, especially because plasmids are closed and PCR only replicates open-ended things, right? Um, but yeah. All right. Hope all is well, and we'll talk